the first appendage we're going to focus on is flagella. So to this point, your knowledge of flagella may only extend to the fact that sperm have a single flagella that allows them to engage in mobility or motility. Flagella have the same overall function in the sense that they are used for movement of bacteria, so they still have that motility function. However, their flagella can be arranged in a different manner compared to what we're used to when we talk about sperm having a single flagella. So with bacteria, they can sometimes have a single flagella. If that is true for a particular bacteria, then we say that that bacteria has monotrichous flagella arrangement. So this bacteria up here would have monotrichous flagella arrangement, and that is noted from a single flagella sticking out of the bacterial cell. Some bacteria have what we call a lophotrichous flagella arrangement, which means that they have numerous flagella originating from the same site. So if we look at this bacteria here, it has numerous flagella, all originating from that same site, so that is considered lophotrichous arrangement. Here it's 3, but it doesn't matter if it's 3, 10, 50, 100. If they're all originating from that same site on the bacterial cell, then it's considered lophotrichous arrangement. Amphotrichous arrangement would be a bacteria that has flagella on both ends of its poles, or both its left and right side. So over here, this is amphotrichous arrangement. We see one flagella over here, one flagella over here. Same here, there's 3 flagella on this side, 3 flagella on the other side. But it typically is going to be an equal number of flagella extending from both ends of the bacterial cell. Then there's paratrichous arrangement. Paratrichous arrangement is when the flagella are randomly dispersed all over the entire circumference of the bacterial cell. So there's no particular order to it, they're just randomly dispersed all over the entire cell. So we know that bacteria have flagella in order to engage in motility or movement. They typically are responding to some type of chemical stimulant, and when this is the case, we say that this movement is chemotaxis. So chemotaxis is the movement of bacteria in response to some type of chemical signal. If it's something that's favorable to them, something that they want to acquire, then we say they're going through positive chemotaxis because they're going to be moving towards it. So, for example, if a bacteria was moving in the direction towards food or nutrients, that would be an example of positive chemotaxis. Sometimes bacteria are moving away from something that could be potentially harmful to them. That would be a case for negative chemotaxis. So in negative chemotaxis, bacteria are moving away from a potentially harmful thing. It could be an antimicrobial drug, such as an antibiotic, or possibly even an immune cell, such as a macrophage or neutrophil, some type of phagocytic cell that could engulf and destroy the bacteria. But the generic term for the movement is called chemotaxis. If they're moving towards something favorable, that's positive chemotaxis. If they're moving away from something potentially harmful to them, that's negative chemotaxis. So with bacteria, they're unique in the sense that they don't have an abundance of ATP or energy at any given time. So when they need to go through chemotaxis, they are often not able to get to where they need to be in one round of movement. So they typically have to go through different stages of kind of moving and not moving in order to get to where they need to be. And these 2 different kind of stages are called runs and tumbles. So during a run, a bacterial cell is going to rotate itself in a counterclockwise direction. This is going to wind the flagella and allow the bacterial cell to take off in a particular direction. So we would classify this as a run. What's going to happen though, likely before it gets to its final destination where it needs to be, it's probably going to run out of energy because bacterial cells don't have an abundance of energy like what we would expect for a eukaryotic cell. So when it runs out of energy, it is actually going to induce something called a tumble. During a tumble, the bacterial cells actually move in the reverse direction, so they're going to rotate clockwise. The flagella untangle, and the bacteria start to free fall in something called a tumble. During this free fall, they're no longer moving in that general direction anymore. During the free fall, they are actually just going to be producing more ATP and kind of building up that reservoir again. Once they have enough energy built back up, then they can go off into another run. So then they'll rotate counterclockwise and they'll run until they use up all their energy. At that point, hopefully they're where they need to be, either away from whatever was trying to get them, or they come in contact with some of the nutrients that they were trying to get. But if they run out of energy before they get to where they need to be, then it's quite simple. Then they just turn clockwise again and induce a tumble and build up their energy reservoir. So typically these bacterial cells will go through a cycle of runs and tumbles in order to engage in motility to get towards something or away from something. But just keep in mind, for them to initiate a run, they have to turn counterclockwise, and that is going to expend a large amount of energy. For them to initiate a tumble, they need to turn clockwise, and that is going to allow them to build energy so that they can eventually take off in another run. And in this image, we have an example of what a bacterial cell would look like if it was moving. So here we're able to see a bacterial cell actually engaging in motion. So we can see the bacterial cells kind of zipping around. This one's kind of stuck in a circular format, but we can see the other ones kind of zipping around through here. And again, that would be them using their flagella. They would be going through cycles of runs and tumbles to kind of use energy and then build their energy at various points. Okay, so the next appendage that we're going to talk about are fimbriae. So fimbriae, they are these short hair-like fibers that extend all around the surface of the bacterial cell. I like to imagine them as the little hairs on a caterpillar. So if the caterpillar is like a rod-shaped bacteria, those little hairs all around the surface of the caterpillar would be the fimbriae. And what the fimbriae are responsible for are attachment to surfaces. So typically, bacteria that have fimbriae will use those fimbriae to attach to surfaces in the body, like cells or tissue, or possibly attach to surfaces in the environment. So, for example, if a bacteria gets into our body and it wants to stay to cause an infection, it will typically have to use its fimbriae to attach to some type of cell or surface in our body, like a tissue. Because if not, our body will find a way to flush it out. We could cough it out, we could sneeze it out, peristalsis can move it through the gastrointestinal system and eventually out. So if we're thinking like a bacteria, if it gets into a human host, it wants to attach to something so it can stay. And that's what it would use its fimbriae for. It would use the fimbriae to attach to a surface, specifically in the human body. Some bacteria have an additional appendage called a pili. The pili, they are used for attachment, but not attachment to surfaces like what we were talking about with fimbriae. Pili are used to attach two bacterial cells to one another for the purpose of communication. So here we have two bacterial cells. You can see they're attached to each other by this long appendage here. This is what we're talking about when we say pili. This is actually a long, hollow tube that bacterial cells can use to exchange genetic information. So I kind of always imagine it as the tube at the bank where you can put your deposit in and it gets sucked across to the teller. Kind of imagine it as that. So typically what happens is this bacterial cell has some type of genetic information that's advantageous. Maybe this bacterial cell knows how to become resistant to an antibiotic, or maybe it knows how to produce a dangerous toxin. So this bacterial cell has that genetic information, and this bacterial cell wants it. So what can happen is this bacterial cell can make a copy of that genetic information and send it across through this hollow tube, through this pili, to this bacterial cell. And now that bacterial cell has that genetic information and can now acquire that trait, whether it's resistance to an antibiotic like penicillin, or being able to produce a toxin like the tetanospasmin toxin that's prevalent for the tetanus bacteria. So this is definitely a way that bacterial cells can learn how to do new things, develop new traits. And this is just to kind of summarize all these different structures. So with what we talked about flagella, and flagella are used in movement. They're not pictured here, but they are used for movement, and they can be present in a variety of different arrangements, and they go through runs and tumbles. With fimbriae, they're these short fibers that are present all along the surface of the bacterial cell, and again, those are used for attachment to surfaces such as cells and tissues within our body. And then here we have the pili, which allows two bacterial cells to attach to each other for the purpose of communicating genetic information. The last appendage external appendage that a bacterial cell can have is the glycalyx. And the glycalyx is more of an outer covering that can surround the entire surface of the bacteria. There's two different types of glycalyxes that can exist, a slime layer and a capsule. So if a bacterium has a glycalyx that is a slime layer, it's typically loosely associated with the cell. So if this is the bacterial cell here, the slime layer is going to be this water-based layer that surrounds the entire surface of the bacterium, but it's going to be very loosely associated, which is why it gets that name of being a slime layer, because it's going to have that kind of watery, flowy type consistency. And what the slime layer does is it protects the cell against dehydration. The slime layer does have a high concentration of water, so if a bacterial cell would find itself in a dry environment without moisture, if it had a slime layer, it could utilize some of the moisture, the water from the slime layer to survive in that dry environment for an extended period of time. Whereas if it got into a dry environment and didn't have a slime layer, it would dehydrate much faster, shrivel up, and die. So a slime layer is typically loosely associated with the cell and helps bacterial cells, it helps protect them against dehydration. The capsule, if a bacterial cell has a glycalyx that's a capsule, capsules are more tightly associated with the bacterial cell. I kind of think of them as the outer covering of like an M&M, so they're more tightly bound to the actual bacterial cell. And what the capsule is going to do is it's going to protect the bacterial cell against phagocytosis. Phagocytosis is our body's primary way to clear bacteria. We have phagocytic cells like macrophages and neutrophils that circulate through our body, through our blood and tissues, and they engulf and destroy bacteria. If a bacterium has a capsule, they are resistant to phagocytosis. So our body is not able to clear those bacteria. Those bacteria persist and continue to cause severe infection, one that often can only be treated with antibiotics because our immune system has a really hard time clearing it because these bacteria are protected by the capsule. They're able to avoid phagocytosis.